Okay. We have X, Y, and Z. This is the X region, this is the Y region, and this is the Z region. Represents a range of possible chemical groups. Now, when we are talking about it, regions X, Y, and Z affect the properties of cell surface membrane in different ways. Which row shows the effect of each region on the properties of cell surface membrane? Let's first talk about R. What is our R? What do you guys think? Sorry, our X. What do you guys think X is? What should I identify as my X? We know the parts of phospholipids. We've studied the structures of phospholipids. What do you think is my X? Any idea, Khadija Aruj, and what my X can possibly be?
A phosphate head?
Yes, this is your phosphate head. Correct. The phosphate head, if you take a look, so this structure that you see over here, this structure is your glycerol. What was glycerol? What did I tell you guys glycerol is? I told you guys glycerol is an alcohol that has 3 OH groups. See, one OH group, it has 3 OH groups. Now, in triglycerides, that is fats, I had told you guys that what happens, that all 3 OH groups are attached to carboxylic acids. Carboxylic acids are your fatty acids. So all 3 OH groups are attached to your carboxylic acids. Over here, the situation is slightly different. Over here, 2 of the OH groups are making a bond with the carboxylic acid. The third OH group is making a bond with our phosphate group. So the phosphate group and this is my alcohol, and combined together we get our phosphate head. So trialcohol, glycerol is a trialcohol, it's got 3 OH groups. In triglycerides, all 3 OH groups are attached to fatty acid chains. In phospholipids, one OH group is attached to the phosphate group. The rest of the OH is attached to your carboxylic acids. And what kind of a bond are we going to be making? This is a bond between alcohols and carboxylic acids, which you studied in chemistry as ester bonds. Right? Now, what is Y and Z? How can I identify Y and Z? What is Y and Z? What should I label it? What do you think? What is Y and what is Z?
Y is saturated hydrocarbon chain and
Yes.
Z is unsaturated.
Yes. Y is your, both of these are basically fatty acids, right? Only that Y is a saturated one and Z is an unsaturated one. But both of these are going to be classified as fatty acids. Now that we have identified X, Y, and Z, we can now try to answer the questions related to it. So they're saying which row shows the effect of each region on the properties of cell surface membrane. So you have to tell which region of the molecule has these properties. The first region is increases permeability of the hydrophobic region. Who do you think of these 3 regions, X, Y, and Z, which one is increasing the permeability of the hydrophobic region? What do you think? Permeability means that it's making it more permeable, so it's allowing more molecules to pass through. More oxygen molecules, more hydrogen molecules, more of your carbon dioxide molecules, right? All of these molecules are small, non-polar molecules. They can cross the cell membrane freely. They can even go to the hydrophobic region. So what region increases the permeability of the hydrophobic region? What do you think? You have the
C.
option of the phosphate head. Say it again.
The Z, the unsaturated fatty acid.
Unsaturated fatty acids, right? So unsaturated fatty acids increase the, uh, as we have studied, what do? They increase the permeability of the membrane and they increase the fluidity of the membrane. That's what we had studied, that they increase the fluidity of the membrane. So that also helps in increasing the permeability. So there's the fatty acid chains are less packed together, they're less condensed together, so they are more likely to, allow the molecules to pass through. Second is repels polar molecule. Which do you think? What part of this repels polar molecules? Polar molecules are water soluble molecules. What part of the phospholipid repels water soluble molecules?
Both the fatty acid chains.
Both the fatty acid chains Y and Z. Both of them are your hydrophobic regions. We call them hydrophobic tails. And as a result, they are both going to be responsible for repelling your water molecules. Finally, they're saying attracts water molecules. Which portion attracts water molecules?
X.
X is going to be attracting your water molecules. So that's your phosphate head. So let's go ahead and take a look at the options that they have provided us. If we look at the options that we have been provided with, so we said that Z is going to increase the permeability of the hydrophobic region. That corresponds to option D. Repels water molecule, both Y and Z, both the hydrophobic chains. This is also correct. And it attracts water molecules, that is X. So our answer over here is option D. Okay. Continuing on with the next question. Which statement about collagen is correct? Okay, not strictly related from my topic, even though you guys have studied proteins with me. So you should be able to answer it. But since it's not our topic, we'll move on to the next question. Which statement about fluid mosaic model of a membrane is correct? The movement of phospholipids in the membrane is reduced if the fatty acid tails are saturated. Glycoproteins in the outer layer of the membrane can move. Channel proteins are fixed in position. Sorry, there we go. What do you guys think should be the answer over here? The movement of phospholipids in the membrane is reduced if the fatty acid chains are saturated. Is that true or is that not true? What do you think? If the acids are saturated, so does that mean that the movement of the phospholipids, what does that mean? What are they talking about? When they're using this term, the movement of phospholipids, when they're using this term, what are they actually trying to say? They are talking about fluidity. The membrane of so they are saying that the fluidity of the membrane will be reduced if, what's the condition for it? They're saying if the fatty acids are saturated. Is that true or not? Would saturated fatty acids affect our membrane fluidity? Would they make our membrane less fluid? Yes or no?
Yes.
It's true. It's true. So if we have a lot of saturated fatty acids, that will make our membrane stiff. So the phospholipids will be able to move considerably lesser. Glycoproteins in the outer layer of the membrane can move. Is that true or is that? What do you guys think? Is this something that is correct? Glycoproteins in the outer layer of the membrane can move. Is that true? Is that something that actually happens?
No.
Okay. When we're going to go ahead and we're going to talk about this is actually true. Glycoproteins can actually move around within the cell membrane. They have the ability to move. This is a fluid mosaic model. There is a reason it's known as a fluid mosaic model. That is because of the fact the components of your the components of your cell membrane have the ability to move. So glycoproteins in the outer layer of the membrane can move. This is actually correct. Finally, they're saying channel proteins are fixed in position. When we are going to go ahead and talk about it, this is incorrect. Channel proteins are not fixed in position. Channel proteins can change their position as well. So our answer over here is going to be only 1 and 2 are correct. So our answer is option B. Okay. After that, the question is saying diagram 1 and diagram 2 show how the transverse section through a leaf changes when the leaf is moved from solution X to solution Y. Which row describes the water potential of leaf cells and surrounding solutions in diagram 2 compared with diagram 1? So what do we think? If we take a look at diagram 1, you can see that the leaf has become curved, right? The leaf has become curved. Now, and if you look at diagram 2, we can see that the leaf is actually quite straight. The leaf is actually quite flattened. Over here, what has happened? The leaf has become flaccid, right? The leaf has become flaccid. What does flaccid mean? Flaccid means that it's wilted, right? Now, when would the leaf become flaccid? The leaf will become flaccid when the water will move out of the leaf into the surrounding solution, right? The water would move out of the leaf in the surrounding solution. Only when the water is leaving the cell, the hydrostatic pressure would decrease and the leaf would become flaccid. It would become smaller. What about over here? Over here, our leaf is becoming flattened. It's becoming turgid, right? When a leaf is becoming turgid, that means that the water must be moving into the leaf. As a result, the water potential inside the leaf is increasing, or the pressure inside the leaf is increasing, and that's the reason your leaf is becoming flat. It's becoming turgid. So, what is going to happen over here? Over here in this diagram, I think we can clearly see that solution X has low water potential and the leaf must have high water potential, right? The leaf must have high water potential and the solution must have low water potential. That is why what's happening that the water is moving from inside the leaf to outside the leaf, from high water potential to low water potential. In the other diagram, if you take a look, what is going to happen? The water is moving into the leaf, which means outside the leaf we must have high water potential. Outside the leaf we must have high water potential. And inside the leaf we must have low water potential. Now, we have to compare two things. We have to compare water potential of leaf cells in diagram 2 compared to water potential of leaf cells in diagram 1, right? Water potential of leaf cells in diagram 2 compared to the water potential of the leaf cells in diagram 1. So in diagram 1, the leaf has high water potential. In diagram 2, the leaf has low water potential, right? Since the leaf has less water potential over here, so it's going to be less negative, right? This is going to be less negative. Over here, since I have a higher water potential within the leaf, it will be more negative because there is more water potential within the leaf. So it's going to be more negative. If we have a low water potential within the leaf, so it's going to be less negative. So diagram 2 is less negative and diagram 1 is more negative. So they're saying compare diagram 2 to diagram 1. So diagram 2 is less negative compared to diagram 1. So our answer is either option A or option B. Then let's take a look at the next part. The next part is stating the water potential of solution Y compared with water potential of solution X. Now, what is happening over here is this, that if we take a look, now we have to look at the solution. So solution Y over here has high water potential, which means that it is going to be more negative, right? While over here, if you look at diagram 1, so we are actually going to end up seeing that this solution has a lower water potential. Since the solution has a lower water potential, so we can see that your solution Y is more negative, solution X is less negative. So our answer over here should be option B. Making sense? Does anybody have any confusion regarding the topic?
No.
All right. Which features must always be present for water to move between two solutions by osmosis? What do you think? This, if you guys remember the definition of if you guys remember the definition of osmosis, this will be really easy. What's the definition of osmosis? Does anybody remember what the definition of osmosis is?
The net movement of free water molecules from high water potential to low water potential down the gradient through a
Yes.
partially permeable membrane.
Yes, through a partially permeable membrane. So what are the requirements in the definition of osmosis? First requirement is that you are going down the water potential gradient, right? High water potential, low water potential. The second requirement is selectively permeable membrane. That's it. Nowhere in the definition have we mentioned the concept of carrier proteins. Nowhere in the definition is it written that it occurs across the cell surface membrane. It can occur through any membrane as long as it is selectively permeable. That's what the definition says. So only 3 and 4 are mentioned in the definition. 1 and 2 are not mentioned in the definition. So 3 and 4 are essential. You need 3 and 4 for osmosis to occur. 1 and 2 can help in osmosis or they can't help in osmosis. It doesn't matter. But 3 and 4 are necessary. When living pancreatic cells are placed in a solution of red stain called neutral lead, the cytoplasm becomes red. The cells are then removed from the solution of neutral red. The red stain in the cytoplasm moves into vesicles, which were exported from the cell, eventually leaving the cell colorless. Which transport mechanism could explain how the red stain entered and left the cells? What do you guys think? Think about it. The cell was placed in a solution of red stain and the cytoplasm became red. After that, the cell was removed from the red solution. Now, I will tell you, you can solve the entire question simply by figuring out how the dye is leaving the cell. How is the dye leaving the cell? They're saying the red stain in the cytoplasm moved into vesicles, which were exported by the cell, eventually leaving the cell colorless. What method is that? I taught you guys multiple methods. I taught you guys diffusion, osmosis, active transport, facilitated diffusion. I taught you guys about endocytosis, exocytosis, pinocytosis. What method is this? Which method are they talking about over here? This method. What is this one? Sorry, say it again.
Exocytosis.
It's exocytosis. So when you form vesicles and those vesicles fuse with the cell surface membrane, right? And allow the chemical compound to leave the cell, that is exocytosis. So the cell, it is leaving the cell with exocytosis, which means that it is either B or it is D. Now we have to figure out how it is going to be entering the cell. Is it entering the cell through diffusion or is entering the cell through osmosis? What do you guys think? Is it entering through diffusion or is it entering through osmosis?
Osmosis.
What is osmosis? How do you define osmosis? How do you define osmosis?
Net movement of free water molecules from high water potential to
Yes.
low.
Yeah. Movement of water molecules. What is moving over here? What is entering the cell over here? Are the water molecules entering the cell over here? Or is the stain entering the cell over here? Right? The stain is the one that's actually entering the cell, not the water molecules. They are not talking about movement of water molecules, they're talking about movement of stain molecules. That is simply we have red stain. It's going from an area of high concentration, that is the solution, to an area of low concentration, that is the cell. So what should be my answer? How are they entering the cell?
B. Diffusion and exocytosis.
Yes, exactly. They're entering through diffusion and they're leaving through exocytosis. Correct. All right. Okay, hold on. Let me open up another question for you guys. 1 second. All right, let's go ahead and take a look. Which roles of cell surface membrane result from the properties of the phospholipids? 1. To allow cytokinesis to occur in mitotic cell division. Now, cytokinesis is a topic that we are going to study, inshallah, when we do our topic of cell division. We'll be doing, I think, the next topic or maybe the topic after that is the topic of mitosis. So, inshallah, we will cover mitosis in then. So, cytokinesis is simply the process by which the cell separates into two. So, when the cell breaks apart and it breaks into two different cells, that is known as cytokinesis. So, that is what cytokinesis actually is. Okay. And how does cytokinesis occur? Cytokinesis occur due to what happens over here is that, think of it this way, that you have, for example, a bag of sand, right? And in that bag of sand, you can take a piece of rope, tie it in the middle, and then you can squeeze it. So when you will squeeze that rope, what's going to happen? The sandbag is going to start to squeeze in the middle. So it will become thin in the middle and it will start to separate into two pieces, right? Or if you take a piece of cheese and you try a string in the middle and you squeeze it, what will happen? The middle portion will get thinner and thinner and thinner, and eventually it will break into two. That is what cytokinesis is. So that's the first thing that they're saying. And we will study the entire details of cytokinesis, inshallah, when we do our next topic, mitotic. The topic of mitosis is, I think, next. So, inshallah, when we do that, you guys will understand what cytokinesis is. Now, to allow entry and exit of oxygen and carbon dioxide. So, to allow entry and exit. So, your oxygen and carbon dioxide are able to enter the cell and they're able to leave the cell. Along with that, to allow phagocytosis of a bacterium into a cell. So they allow the phagocytosis to occur. I will, when I go ahead and talk about it, I will let you know that number one is correct. The reason I'm telling you guys number one is correct is because you've not done cytokinesis. I don't expect you to know the answer. Number one, I'm telling you right now is correct. Let's take a look at number two. To allow entry and exit of oxygen and carbon dioxide. Is it a role of cell? So you don't just need to think about, oh, is this what cell membrane does? That's not the only thing you need to think of. You need to think of, is it a role of cell surface membrane a result from the properties of phospholipids? So if the cell membrane is doing this, is it doing because of the phospholipids? Or is it some other molecule? Is it some other component? For example, if your cell, for example, is allowing glucose to be transported, so glucose transport doesn't have anything to do with phospholipids. Yes, the cell membrane will allow glucose to cross, but not because of the phospholipids, because of the carrier proteins, right? That's so because of facilitated transport. So over here, the question is two-ended. Not only are they asking you is it the role of cell surface membrane, but they're asking you, is it the role of cell surface membrane because of phospholipids? So number two, is number two correct or incorrect? The point number one is correct, which I'll let you guys know because you guys have not done cytokinesis, so I don't expect you to know about it. What about point two? Do the phospholipids, not just the cell membrane, do the phospholipids allow entry and exit of oxygen, and carbon dioxide? Yes or no?
Yes.
Yes, this is true. That is exactly what they do. The phospholipids allow the oxygen to enter and exit because oxygen and carbon dioxide are small, non-polar molecules, meaning that they can easily cross the hydrophobic region, so they can directly diffuse in and out of cell. What about the last part? To allow the phagocytosis of a bacterium into a cell, do they do that? Do they allow the bacterium to perform phagocytosis? Keep in mind, I'm not asking about cell membrane, I'm asking about phospholipids.
Yes.
No. The phagocytosis is actually not a function of phospholipids. It is a function of cell membrane, but not specifically phospholipids. Remember, on the cell membrane, what do we have? We have glycoproteins and glycolipids. What was one of the uses of glycoproteins and glycolipids? I told you guys different functions of glycoproteins and glycolipids. One of the important functions of your glycoproteins and glycolipids were that they act as antigens. What were antigens? They act as antigens, they act as receptors. And this is how they help in immune response. So it's actually the glycoproteins that allow that to happen. The glycoproteins and the glycolipids help in the immune response, and they allow the phagocytosis to occur. So over here, only 1 and 2 are actually correct, which means our answer will be option B. After that, they're saying which factors can be changed to affect the rate of facilitated diffusion. Which factors can be changed? Which factors can be changed to affect the rate of facilitated diffusion? What do you think? Which factors can be changed to affect the rate of facilitated diffusion? First is surface area of the membrane. If I increase or decrease the surface area of the membrane, my facilitated diffusion, will it increase or will it decrease, or will nothing happen to it? If I increase the surface area of the cell membrane, will it help diffusion, facilitated diffusion, or will it have no effect? There will be no change. It will remain exactly the same. What do you guys think? Yes or no? If I increase the area of the cell surface membrane, think about it this way. If I increase the area of my cell surface area of the membrane, if I increase the surface area, will the facilitated diffusion increase? Will I see more diffusion or less diffusion? Or will there be no change? Is it going to increase or is there going to be no change? What are you going to see?
It will increase.
It will be increased. That's right. So surface area of the membrane will increase the facilitated diffusion. The surface area will affect the facilitated diffusion. What about concentration gradient? If you increase the concentration gradient, will that increase the diffusion or would that bring no change?
It would also increase the diffusion.
It will also increase the rate of diffusion. The number of specific protein channels. If you increase the number of protein channels, will that increase facilitated diffusion or will there be less or will there be no change whatsoever? This is
The
also, yeah,
yeah,
go on, say it.
it's also true because it will
Yes.
also affect.
Yes, your facilitated diffusion, what was facilitated diffusion? When particles were diffusing with the help of protein channels, right? That was the problem with it. That The problem was that the molecules could not diffuse directly through the cell surface membrane. As a result, they needed the help of protein channels to help them through. So, greater the number of protein channels, the faster your rate of diffusion is going to be. So, our answer is option B. Option A. All of these options are correct. Next, they're saying the diagram shows an experiment using a model cell to investigate the movement of substances. So over here, they have set up an experiment. So for a cell, to show a cell, they have taken this tube. This tube is generally known as a whisking tube. A whisking tube acts like a partially permeable membrane. So it acts like a cell membrane. Okay, so we have to think about it like a cell, my cell. Okay, this is my cell. The cytoplasm is inside of it, and the lining or the wall is going to be acting as my partially permeable cell membrane. Now, they're saying we have a membrane. This membrane is permeable to monosaccharides. Monosaccharides are glucose, fructose, galactose. It's permeable to monosaccharides and it is permeable to water. So that's what we're going to go ahead and notice, that this is permeable to monosaccharides and water. Then on the outside, I have a solution that has 0.01 mole DM cube sucrose and 0.01 mole DM cube glucose. Inside of the cell, I have 0.03 mole DM cube sucrose and 0.02 mole DM cube glucose. So if we take a look, sucrose is higher within the cell, right? Sucrose is higher within the cell, right? And it's lower outside the cell. Glucose is also higher within the cell and lower outside the cell. The difference in concentration between sucrose inside the cell and outside the cell. This is inside and this is outside. Now, between sucrose, if you take a look, so inside the cell and outside the cell, the difference is greater, right? Greater difference in concentration. Between, if you compare glucose, the difference is lesser, right? Because over here, the difference is 0.03 minus 0.01, 0.02. Over here, the difference is 0.02 minus 0.01, so that's 0.01. So over here, the difference is much larger. Over here, the difference is much, much smaller, right? So this is basically what you end up seeing. Now, which statements are correct? There is net movement of sucrose out of the model cell. Is this true or not? Is sucrose going to be moving out of the cell or is it going to stay within the cell? What do you guys think? Is sucrose going to diffuse out of the cell or is it going to stay within the cell?
Out of the cell.
Okay, I will tell you you are incorrect. And if you can tell me why you're incorrect. If you can figure out why you're incorrect, if you can figure out why sucrose is not moving out of the cell, I'll be very proud of you. If you can figure out why sucrose not moving out of the cell, because sucrose should move out of the cell. Because inside the cell, the concentration is high, it's 0.03. Outside the cell, it is low, 0.01. Why? Why is it not moving out of the cell? Can anyone figure it out? Why is it not going to move out of the cell? It should move out of the cell. It should be moving out of the cell, but it doesn't. Why? Whoever can figure out, it's a huge thumbs up from me. If they can figure out why is sucrose not moving out of the cell, the answer is written in front of you, just so you know. Anybody? Khadija, Aruj, any idea why sucrose is not going to move out of the cell? It should move out of the cell because diffusion, but it won't. Why? Okay, I'll tell you guys why. And I highlighted it for you guys as well. It was highlighted over here. Do you guys remember how I highlighted membrane? How I had highlighted for you guys membrane permeable to monosaccharides and water. What are the monosaccharides? The monosaccharides are, we had studied that there can be trioses, right? You can have riboses in which you had deoxyribose, right? You had your ribose sugar. We can have our hexoses. In hexoses, what did we have? In hexoses, we had glucose, we had galactose, and we had fructose, right? What is sucrose? Sucrose is a disaccharide. We studied three disaccharides. We studied lactose, sucrose, and maltose. So sucrose is a disaccharide. That is why it's not going to be moving out of the cell because the cell membrane is not permeable to disaccharides. It is only permeable to monosaccharides, meaning that only glucose, galactose, and fructose will be able to actually move out of the cell. Other than these, other than your deoxyribose, other than your ribose, other than your trioses, other than the monosaccharides, nothing else will be able to move out. So sucrose is a disaccharide. That is why it's not going to be moving out of the cell. So if the this did not say monosaccharide, if it did not say monosaccharide, if it simply said carbohydrates, then sucrose could move out. But they specified, and they specified for a reason, and I highlighted it for a reason. The reason is monosaccharides. Sucrose is not a monosaccharide, hence it cannot cross the cell membrane. After that, they're saying there is net movement of glucose out of the model cell. Is that true? Is glucose going to be able to move out of the cell?
Yes.
That is true. That is correct. Finally, they're saying there's net movement of water into the model cell. So is the water going to move into the cell? Is the water going to move into the cell? Where is the water going to move? Is the water going to move?
Yes.
Yes, the water is going to move into the cell because inside the cell we have a lot of sucrose and glucose. We have a lot of solute, right? We studied the topic, we talked about solute potential and solvent potential. So we have a lot of solute. Since we have a lot of solute, so the water potential inside the cell is low. So water will move from outside the cell to the inside of the cell. Hence, our answer over here is option D, 2 and 3 are correct. Hold on. Just okay, Khadija will go ahead and we will stop it over here for today. And